That's what I have the YouTube Premium account for, so they can dump whatever file we get there, 4 GB. Um, let's see. Okay. And since I'm recording, might as well start punctually. What do you say? It's not all of us here reunited. I don't know how many of us are Feyenoord hooligans. With a small group, this type of review is going to miss a bit of the juice of interaction, but I wouldn't be surprised if the last minute online just made everybody think, well, we're going to be recording this. There's no need for me to participate. I'll just enjoy it all offline in the comfort of my own sleep, for example. Any other reasons for me to be imagining that we're not going to be fully fledged today? So how about this? Just activate the knowledge a little before we go further. And it's just between the few of you. And some of these answers, you have it from last week. Some of them you will be thinking about a little bit, and we're going to discuss them just to cast a little bit of a wider spectrum on what a tuning curve may be and the properties it may have. I am presuming that you can see the Mentimeter screen I'm sharing. Can you?
Yes, we can.
Very good. It was a fun experience teaching during quarantine because could record a lot of lectures and it was convenient. But I gotta say that this is the first online lecture I'm having in quite a long time, and I'm reminded how odd it feels. It's so much better to be in the same room with everybody at the same time. For the latecomers amongst you, the reason why we have 3 video feeds is because I've asked everybody to have a video feed if their bandwidth allows. So if you'd be so kind as to share your semblance to us, then I can estimate how much of the information has been transferred. Bring in the videos. Thanks, Victor. We have 5 responders and 20 people online, so this is not exactly, I don't know, 20 really. What, 10, 12, including me? It's about 50%. I'll move forward when we have 8 responders. Five responders is a good number, but insufficient. So if I'm wondering, not many more people have joined in the video. That most likely signifies that they fired up the Zoom and are not there. Hi, Hinske, nice to see you. Lucy, Have you seen that we have a Mentimeter online? Have you guys answered already? I said that I was going to, yeah, I'm going to move forward when we have 8 people. So while you're answering the Mentimeter, I'll let you know what the day is going to look like today. After this little 2 Mentimeter slides, some about the tuning curve, some about the 3 blue, 1 brown homework assignment, which you had only 2 days to accomplish given this odd week. Then we're going to play in the playground together. It's not raining in Breda, so online playground that is, in which we are going to be concocting a feed-forward neural network. We're going to have a group discussion after that. And then I'm going to give you some extra aspects of hierarchical neural networks, and I'm going to be telling you about how you slice hyperplanes. This is based on some observations that I'm hoping you're going to be building through the TensorFlow playground that we're going to be playing with. And also I'm going to lay on you some essentials of computational neuroscience relating to invariances and interesting neurons with very sophisticated receptive fields. And then finally, we're going to be talking about learning and plasticity, which actually is the main topic of the lecture today, main topic, the guiding topic, let's say. Let's see how many responders we now have. 7. Wow, we're getting there, we're getting there. Greetings, Boris. Thanks for your video. So yes, I am presuming that, let's see, we have now at least 8 people, don't we? 9 people actually. So 8, excellent. So we move forward. If you joined late, you can still respond, and I will be reacting to the dynamic changes in these scores. The tuning curves. It measures the response of a neuron, and you're all convinced that that's what it does, but it actually measures the response of a neuron to a particular stimulus. So without that specification, this question is a little bit ambiguous. I'm glad that you pattern completed this to represent what it truly is. As you remember, the tuning curve on the x-axis has a stimulus, and on the y-axis, generally a firing frequency, can be other types of activity, of neuronal activity. And that's why you strongly agree that the tuning curve measures the response of a neuron. Now, the vast majority of you intuited that is not exclusively a Gaussian function. Again, this is a question that I was kind of hoping that would divide opinions, because and in a sense it does. So we do have 3 people that, the 6 said it is not a Gaussian function, 7 even. And I'm going to now ask, if you are one of these members of the 7 here, can you tell me why you strongly disagree that a tuning curve is a Gaussian function? I'm basically asking for a volunteer.
I think in theory it could be any kind of function, right?
It could be any kind of function. I mean, especially because since you're plotting simply the activity of a neuron against some stimulus you give, there's no guarantee that the stimulus you give is going to produce a particularly well-organized function. The Gaussian function is natural when you have some sort of radial base, such as, for example, the orientation of a bar, and then it's radial because 0 degrees is equals to 360 degrees. And if you actually have a clear preference, then you're going to have a single maximum, and is going to decrease the activity around it. So one of the ones that I shown you was actually really did look like a Gaussian. But in the case of Dango, many of you have noticed that it's not necessarily a Gaussian. You have also tuning curves that look like a sigmoid. And those are very common when we are dealing with stimuli that end in an interval, say, for example, brightness. And so, the brighter it gets, the more it responds, the darker it gets, the less it responds. Now you can have tuning curves for very complex stimuli, say for example, olfactory stimuli. You'd have, say, taste of strawberries or smell of strawberries and smell of bananas. It is weird when one starts to put all of the imaginable stimuli, and especially if they're complex, on the x-axis of something. In fact, it turns out that most neurons are multimodal, and even orientation neurons, there's a paper in Science in 2021 showing that the neurons in the visual cortex that I told you that respond only to orientation actually co-encode orientation and color. And so you must think not only about a single curve as a tuning curve, but as a tuning surface, right? So on one dimension you have color, and the other dimension you have orientation. And it gets more complex the more stimuli you can produce to your neuron. So this is just for you to get the sense of how complex a tuning curve can become and how impressive it is that the Nobel Prize has been, the reason why the Nobel Prize has been awarded to that discovery is because it is a simple way to start. I mean, like you just have like an orientation selectivity, and that's a tuning function. Then we move on to relates input to frequency. That is unison. Yes, it does, particularly if you're measuring single neuronal firings. You could also, for example, be measuring blood oxygenation levels in an area of the brain. And in that case, the tuning curve would be looking into oxygenation levels as a function of the stimulus. So ultimately, it is a tuning to that the neuronal population is going to give you, the activity of the neuronal population, of which firing frequency is but a proxy. So you have many more types of response functions as such. Most of you strongly agree that it should be continuous, and it makes sense, right? Well, what makes it continuous is because you can always record that function along the dimension of stimulus you've produced. So if the dimension of stimulus is continuous, the neuron, either fires or doesn't fire, but it always has a response to the stimulus. It could even be a noisy response. It could actually do something else every single time. The variance is going to be great, but ultimately it is a continuous function because of the way it's defined. It can be a single neural property. Usually we think about it, the tuning curve, as a single neuron property, but you can also talk about tuning curves of areas, and it depends really on what you're measuring. But the way in which it's discovered first was as a single neuron. That's why you flatten out. That's lovely. That's quite a nice feedback here. I noticed that there's some points in the chat. Let me open the chat and see. Oh, the Mentimeter code. Oh, thanks. Okay. Now is unimodal. That's also great. I mean, you guys are on the mark with this. So you neither agree nor disagree. Sometimes unimodal, sometimes it's multimodal. It really depends on how cleanly that neuron represents that property. Tendentially, the closer to sensory interfaces, the more unimodal the tuning curve will be. And obviously it can be multidimensional. So, you're nanobiologists, you're like multidimensional thinkers. So multidimensional doesn't scare you, which is really nice. Now to move on, thank you for this. Any questions about tuning curves in general? The review of the Nango project is basically this slide. We are going to assemble a little bit further. If you have questions about the project, you will be able to use some time in a moment. When you're working in the playground, you can also have a one-on-one with me to ask specific questions about the playground. I'm not going to give like a full-fledged review, given that we already had the demo last week. Next slide says, how much of the homework did you do, and what kind of boost that gave to your understanding? Now, we 3 Blue 1 Brown was the idea that we're going to be looking into artificial neural networks and the small beginnings of feed-forward neural networks or deep neural networks, whatever it is that we're calling them these days. If you didn't watch it, the whole lecture today might feel a bit floaty. So maybe during the time that we do our playground thing, might be an idea to leave it playing in the background. They're very fundamental. Grant Sanderson, the head of Free Blue and Brown, is possibly, I mean, I don't need to tell you about him, but he is one of the greatest educators in math right now alive, I believe. That's all right. So we have a little bit of a spread. Now, clearly, if you didn't watch, I see that 3 didn't watch the videos, that would mean that all of the rest of the information is locked away from you. I understand the constraints of life, so I'm not going to punish you for this, but I will use this moment to recommend do watch it. And what we're going to be doing today, after we talk about this, so we're going to discuss a few elements of this. I just wanted to get a sense of what is the resonance with the nanobiology team. So thank you for answering this one. This is exactly what I wanted to know. Many of you actually watched it. I presume that those who did not watch it for lack of interest, but for lack of time. So you're going to get the chance now. I also know how hard it was, like from Friday to Monday. That's I had the same problem in preparing this lecture. Now, moving on, we're now going to actually deploy some of the insights that we had in 3 Blue and Brown via the TensorFlow playground. I need to find another window from Brightspace. So if you go to content, and if you go to lecture 4, there is something that's called the playground. Yes, so it was, so Boris said that you missed that you needed to watch them. Well, it's called homework in Brightspace. I thought that I did mention it and I called it homework, but alas, I mentioned it a couple of times, but maybe I didn't really underscore it or create an announcement. That's also on me then. Apologies, Boris. In any case, now what I would like you to do is, I think that you'll be fine anyways. You can watch it later. These materials are modularly sufficient that even if you didn't watch them ahead of time, I think you will find yourself jumping in quickly into this. But this is what I wanted. This is the activity that I had planned for us today. Click on TensorFlow Playground Quiz, and this is a PDF that explains you a few ideas and then points you to the TensorFlow Playground. And here it is. So the TensorFlow Playground is a feed-forward artificial neural network that you'd be training via backpropagation by pressing play, for example. And you can select different data sets here. And you can try to, via gradient descent, discover the weights that will maximize or minimize the error, maximize classification accuracy of these two data sets. You should read should read a PDF, like to get a sense of the instructions, and I'm going to break out rooms. You can self-assign the room you want, okay? So I'm going to break out like 5 rooms so that you can self-assign, and then you can call me in for us to discuss these ideas. I just wanted to introduce this to you first. There's a chat question. I'll actually open this on some. What's the importance behind a feed-forward neural network versus not? We're going to talk about hierarchies in a moment, but this is, if you remember in Nango, we've been using neural networks that would feed forward, spiking feed-forward neural networks with specific response functions. I will fry you one slide before you're actually going to go into this very deeply. But Callan, this is a good question. Hold it on. We're going to talk about hierarchies in a moment, and later we're going to talk about recurrent neural networks. It's going to be one of the latest in the latest moments. Now, in any case, here you have a few things to be aware of. If you watched the video by 3 Blue 1 Brown, this would be more natural. If you didn't, this will be more unnatural. These are not number digits recognitions. This is simply a data set for a two-dimensional problem. Now, this is an artificial neural network, and you should think of each of these little dots here on this output as in an input space between two dimensions, x1 and x2. Now, what does it mean from the perspective of brains? What we are trying to abstract away are the particulars of these stimulus dimensions. But what you could be thinking about are, for example, okay, so it's a color and an orientation, or it is any continuous dimension such as loudness, brightness, orientation, anything you can come up with would be x1, so the horizontal, for example, and x2, the vertical. And every point is one stimulus, right? So the bright at this angle, for example. And see the feature here, x1 is giving you large values for data points that are highly horizontal. So now you see this underlying color map. It is a measure of horizontality or of left and rightness. So rights are blues and lefts are oranges. And so every data point, every stimulus is a combination of these two numbers. And what you can then do is train the neural network by pressing play, which will perform the gradient descent and backpropagation into the weights. But interestingly enough, you can also create more layers, and you can add and play with the number of neurons of the layers. And what we're going to be discussing later are going to be the receptive fields of these neurons. So for the moment, don't start yet. I'm just telling you what we're going to do, okay? So this is the TensorFlow playground. Before we go in, I'm going to tell you the general idea behind hierarchies and invariances, as Callan just brought us to. Allow me to find my slides. One of the earliest notions of artificial neural networks, brain artificial neural networks, was the neuron by McCulloch. McCulloch and Pitts were the creators of the first artificial neural network. It is a very simple thing. It is one unit that has one output and two units as input. Is this the right slide? Oh, that's wait. I have multiple slides, so turns out this is not the one I was going for. Wait a second. Here. I'll be transmitting this. So, and this first analogy by McCulloch and Pitts was that neurons seem to be rigged to be able to do some logical operations. I hinted on that when we discussed Nango by saying that we can tune any neural network to produce any function. But what you should be thinking about is the following. Say I have 3 neurons, 2 input neurons and an output neuron. The 2 input neurons synapse with excitatory synapses onto the downstream neuron. And the downstream neuron has a certain amount of current before it spikes. It's a leaky integrated fire, for example, neuron, or it is an on or off neuron that will say on if sufficiently many inputs are coming to it. So if you think about these two presynaptic inputs to the third neuron as frequencies of excitatory postsynaptic currents, then the neuron as an integrator is summing the two sources and applying a threshold, producing a frequency on the other end. Now think about this for a second. The frequency of input can be represented as a number between 0 and 1, from minimum excitatory postsynaptic current to maximum excitatory postsynaptic current for both of the input neurons. And the output neuron is producing a frequency, which is also a number between 0 and 1. If the two inputs are firing maximally, and we set the threshold appropriately, the third neuron is going to fire. This is called an AND operation. Make sense? In logics, we say that A and B obtain if A is true and B is true. So that's a logical operation. Now, this was, people were high on logics in 1943 because computers were just being invented, and McCulloch and Pitts came up with some analogies of what would happen, if we interpreted artificial or biological neural networks as these logical units. And so this is the paper in 1943 that said, because of the all-known character of nerves activity, we can treat them by names by the means of propositional logic. It's an incredibly powerful idea, and it is underlying all the artificial neural network developments now. So back then they were still engineering these solutions and thinking, okay, which structures of neurons of networks are going to give rise to this? But they thought about, okay, so if this neuron excites and this neuron inhibits, maybe we get an OR function, or maybe we get a NOT function. So if the neuron here is inhibiting the other neuron, then we're saying no to the other one. So they were thinking about, how to construct complex propositions of the type that we construct. It's like, if A and B, but not C, and A and B and C not, then D. So these propositional logical statements are abstract versions of the ways in which we think. If Feyenoord is playing and Feyenoord wins, and today is the day after, we don't go to school. So we create these compositional ideas and try to resolve them to come to conclusions. Now, it's interesting because it starts very small at the level of single neurons, but it goes all the way to us. So we analogize. So McCulloch and Pitts, biologist plus mathematician, came up with this idea. And what they then did, they made these following assumptions, many of which actually still hold for leaky and integrated fire neurons. Activity of neurons all unknown. Certain number of synapses must be excited within the period of addition to excite a postsynaptic neuron after threshold. There is synaptic delay. Inhibitory synapses shunt other neurons, which is actually quite true. It's something that we didn't have the opportunity to go very deeply into, but it turns out that inhibition is more effective than excitation, especially if inhibitory terminals come close to the soma. It shunts the possibility of a neuron to produce spikes. And importantly, the weird one here, the structure of the net does not change with time. We're going to make these assumptions. They don't say that it's actually they're good assumptions. Particularly the last one is the idea that, if you want to produce a certain logical function, then the structure of the network doesn't change over time. Turns out that you can change the network over time so that a certain logical function is produced. This is exciting. Now, this is where it all begins. And what this analogizes to is the modern artificial intelligence single abstracted unit. We talked about firing neurons today, or we talked about firing neurons previously primarily, and now we're just distilling the juice of this by looking into the inputs as numbers representing, for example, frequencies, with a certain saturating nonlinearity is a function that is mapping inputs to outputs and producing a number as an output representing frequency that can be fed into other neurons. So this is basically, all there is to the individual processing unit of artificial neural networks today. This is what we're going to be using in the TensorFlow playground. Before I love you and leave you with the TensorFlow playground itself, I want to let you know that the neuron, like as such, this neuron is a linear classifier. And what does a linear classifier do? Well, it says yes or no to something. Now let's think about this as in the terms of linear algebra. Say now you have two types of things, males and females. You have fruits and vegetables, and you try to sort them out. Fruits and vegetables are fun because then you can sort them by, say, shape and color, and you put the shape on one axis and the color on another, and you then have like a data set of fruits and veggies, and then you ask, okay, is it sufficient for me to distinguish fruits and vegetables based on shape and color, say symmetry and color, for example? In this two-dimensional space that is formed by this data, you can then potentially create a line to distinguish them if they are actually linearly separable. Now, it might be that fruits and vegetables are not linearly separable because tomatoes, but it might also be that, to a large extent, you're going to have a lot of good separation. Greens go here, reds go here, and then there's a little bit of stuff going mixed in the middle. I don't know. But the point here is that you can't create a line, or in two dimensions, one dimension is a separating hyperplane, that represents the linear combination between the properties of such that you create maximum separation between the two categories. This may or may not be possible, but this is the problem. So let's think about the neuron as a linear separator. It's going to say yes to certain things, and he's going to say no to certain things, and you're going to create this yes or no function by tuning the weights of the network appropriately. And it turns out that the weights of the network are this hyperplane. Trust that 3 Blue 1 Brown made a very good point of this, so I'm not going to drill on this. But what I'm going to say is that once you are assembling multiple hyperplanes, you're actually creating compositions of these linear functions. And this is what you're going to see in the TensorFlow playground that now you're going to be grinding on. So we're going to look into hierarchical neural networks, and we're going to be talking about slicing these hyperplanes as we combine the activity of different units. And the idea here is that you interpret these problems. Notice, seeing some of these problems, for example, this problem here, with this data set, the oranges and the blues, this is a so-called XOR problem, and they're not linearly separable. So because they're not linearly separable, you can't find one single hyperplane that distinguishes them. You'll have to create another hidden layer. I challenge you to think about this problem as you're solving the questions that I had produced here in Brightspace. So in the document in Brightspace, and for some reason now the document is not coming up. You will find an assortment of questions. When I break down the room, you read this out and you start thinking about it, and then we're going to have a nice little discussion about it. And I think it's a really fun way to basically be thrown into the deep end of the pool of machine learning with respect to deep feed-forward neural networks. You can use ChatGPT if you have questions that you do not know the answer for. You can use your colleagues, and that's the way that I had planned to do this, okay? So you will interact with the AI, or you will interact with me, and we're going to clear up. And I don't think you're going to have the chance to do all of these questions. Maybe some of you will. But, you know, read through, play with the playground, and then in 45 minutes from now, we reconvene. Is that clear? You can take pauses as you like, okay? So now I'm gonna break out the rooms. You can go into the rooms as you see fit. You see who is in the room and, just drop in. I'm going to open, I think, 4 rooms, seems, well, let's see, we have how many people? 14, no, 4 rooms is too much. I'm going to open 3 rooms. And for that time, you're just gonna go through and hopefully there will be enough knowledge and ignorance so that there can be cross-pollination between the likes of you. And then afterwards, we're gonna have a discussion about what we've learned. How does that sound? All right? Questions? If not, then I'm firing up the breakout rooms, and in say 45 minutes, 40 minutes, I'll give you close the works, and then I'll bring you back to the main hall. Ask me questions, I'll be here. Okay? I'll join the breakout rooms and we can then discuss specifics. Okay? If there are no questions for breakout rooms, create 1, 2, why is it not changing? 4. Okay. Let participants choose, okay? And then you can cluster up as you like. Enjoy the process. I hope that this is going to be as fun to you as it will be for me discussing it afterwards. See you in a moment. Rooms are open. If you cannot self-assign, you can also ask me to assign you.
When we put regularization, then we get much sharper hexagon, and otherwise it's more of a blob. Then yeah.
Interesting. So, do you want to share your screen and show us what you got? Anyone has their hexagon on?
We were also thinking about the hexagon thing, that because regularization is making the network more simpler, that apparently polygons are a more simple thing than blobs.
Well, really not really. So in order to get sharp edges, you need the rectified linear units. That's the only way to get sharp edges in the hexagon. If you use the hyperbolic tangent or the sigmoid, you're going to get rounded edges. I'll show you why in a moment when we talk about transformations, and to try to understand the level of the receptive fields here. Because when we say we add the number of hidden units, and we say we want a nice little data set like this. With no inputs, then obviously nothing will be trained. So that's a blob with a hyperbolic. Are you seeing what I'm sharing?
Yes.
So if I change it to rectified linear units, I get a perfect hexagon, because the boundaries are sharp. They're not smooth like in a hyperbolic tangent. Let's chat about that in a moment and why it goes from one to the other. But the thing that you'd like to understand here is that each of these edges are the hyperplanes that are defined by the weights that are projecting downstream. Is this clear? Like that every time that you have combinations between 2 neurons and a downstream neuron, effectively you're creating a decision boundary, or a slicing hyperplane. And so this is a combinatorial process, right? So here, what we, in fact, let's get one last unit. It would be relevant to think about the emergence of these separating hyperplanes. Effectively, what you're looking here in the hidden layers are receptive fields, if you like, are the things for which the neuron are active, respond strongly, and stuff for which the neuron doesn't respond. In the case of the ReLU, rectified linear unit, values to the left are 0, values to the right are positive. Now, we haven't talked about the transfer functions much yet, and I'm going to bring them now. And I wanted you to first play and ask, but were you able to discover what the transfer functions were quickly? I mean, was anyone had problems there to discover what the transfer functions were? Good. There's some chat things too. Is there any, Callan asked, is there any situation where just maxing out hidden layers in neurons leads to worse classification performance? Obviously, it makes training run slower, but assuming you have the compute to run it, feels like free lunch. This is an interesting question, and it's a question that in general is best answered for a machine learning course. I wanted to just point out that the way in which we're thinking about this is slightly still from a biological perspective. However, the point here is really clear. I mean, the more units and the more ways in your slicing your input data set, the more overfitting you might be getting. So say, for instance, now you have a complex data set like the spirals, and you make the problem hard by not adding any of the other features. Now, look at what happened when you just go broke for a bunch of hidden layers and neurons. Now, clearly, it will still. Actually, that's too much. Let's just show that we can have a bit of an idea first. So you have a few problems. First, you make the gradient descent really hard. So it takes longer for you to train the network. Second, the combination between the individual receptive fields becomes hard to make. So you have way more tunable parameters. So if imagining infinite computing power, you're eventually going to get to a solution, but you're gonna suffer a lot in the process. And in fact, here this is what you see happening. The network has multiple minima. They are local and they're competing. And so you have this type of flickering thing. The network is not resolving. How is it going to solve the next problem? So it gets stuck. And there you might need some regularization to simplify the network over time. But one thing is interesting. Did you notice here? Okay, now we have something of a solution, right? So the blue is mostly in the blue and the yellow is mostly in the yellow. So you're basically creating a spiral, but it's not generalizing at all. It's just creating islands of data points that for which the function obtains. So it is not an intelligent solution. It's not trying to model the effect of the data in ways that are intuitive to us, which would be, say, extrapolating spirals. And in fact, it's just trying to create some receptive fields that are creating little islands. And you can see here, the receptive fields on the lower level layers, they are just clustering around a few data points at a time. And the overall classification boundary is a multiplication, basically, of these here, a combination, right? The way in which you create these boundaries is more intuitive if you start slow. So if you started already by going like deep and throwing a lot of neurons, then you very quickly this becomes unintuitive and hard to see where it's coming from. So I definitely suggest that if you're using this as a personal training tool to understand the emergence of separation boundaries in classification problems, do it one thing at a time. Let me point a few things out. Let's look here at the output. Do you guys know how the output is? So the test loss and the training loss mean? Anyone would like to ask a question there? Good. Now you see that the training loss and the test loss particularly, is very noisy. This is an indication of overfitting. You chose weights that do well on your training data, but they don't generalize well. So every time that you test something, they basically oscillate, and you continue to have a big test loss all throughout. So it's very noisy test loss, and that has to do with the fact that you have too many units for a relatively simple problem. Now, another something that is interesting to notice is the time course of this loss function. See here the kink? Suddenly, a generalization occurs, and the network became better at improving quickly. So the gradient descent was flatlining, and then suddenly it dropped sharply. These are the kinds of things that you expect in training problems like this. Now, when you have a very big data set, like the brain's acquiring a lot of data set, it might very well be that you do not get your insight, that you do not suddenly, so this is basically an epiphany, if you like. It is when the network discovered, I should do this, and then will make my learning problem easier. It's a little bit like learning numbers when you're 5. And so the error surface is relatively complex for problems like this. But the reason why I wanted to bring you to this is for you to think about receptive fields as they evolve in the artificial neural networks as in combinations. This is the main reason why you're doing this. It's not so much to think about the machine learning problem, but it's more about thinking like the hierarchical feed-forward neural networks. And that's what we're going to go into in a moment from now. Did you ask more questions in the Mentimeter or here? Okay, now we're done with one of the rooms. How about the second room, guys? I mean, 2 questions, 2 observations. I made a remark in the Mentimeter. I'm getting there. I think the amount of neurons should never increase per hidden layer. Okay, that's an interesting observation, Luke. Why would you say so? Can you why would you say?
Yeah, I didn't really test it. It was more something that kind of felt intuitive, because of it, I feel like you should be able to perform the same thing. But yeah, like you would get the same effect without the extra hidden layers, because they're not condensing more information. They're just, well, applying more math that could have been done in one step.
That's right. I mean, so this is truly I think one of the essences of the problem. First, you get the sense that, it's a lot about intuition, right? So like, how would you go in and formalizing this intuition? Well, the complex decision boundary is going to represent more than what you want it to represent. So it has to match the complexity of the problem. In general, it's a good idea that you match the complexity of the problem. These are all relatively simple problems. So too many units are only going to make the decision surface very rigged, overfitted, complex, hard to interpret, bad to generalize. So all shitty things. But it doesn't mean that you don't need many neurons to deal with a bigger data set where you don't have, say, only 2 input dimensions. Say if you have a picture of 256 pixels, those are 256 dimensions, right? And so for more complex input spaces, it is relevant to have more neurons that combine, say, for example, local neighborhoods. And this type of stuff also was transmitted in the 3 blue 1 brown problem. Very good observation, thank you. Other observations? Other remarks? Questions?
I did have one more question or remark. It's not necessarily related to this, but more to the 3 blue 1 brown videos. So in the videos, they try to analyze a picture or a number and see if a neural network could detect what video or what number that was. And I'm wondering, how would you use a different amount of hidden layers to make sure that the image could be in any position? So that was
Great question.
Yes.
Great question, sir. This has to do with translation and scale invariance. Translation, rotation, and scale invariance. I have a few slides about that. You don't need too many more layers. I think that I'm going to get there now. The plan was that after this discussion, we talk about hierarchical neural networks and what they can do. We're going to get to that. But I will tell you, since you ask, in 1977, a Japanese researcher called Fukushima, just like the power plant that exploded, he came up with an architecture called the neocognitron. And it was the first deep network. And that's precisely the problem that he was solving. So the idea to have position invariance. Ultimately, you solve that with convolutional layers, intermediate convolutional layers. You don't need many more, but the more you know about your problem, the better. So I think that here we have two intermediate layers in the 3 blue 1 brown videos. I think one more would be sufficient, but it would have to have specific topology. So you need to cover specific areas of the visual field at a time. Remember when we discussed last lesson, we talked about topography of neural networks and how interesting it was that the retina was topographically mapping the outside world, and this topographical mapping was transmitted to all the other layers, the retinal ganglion cells, the lateral geniculate nucleus, the visual cortex number 1, the occipital cortex, and also the other layers of the visual cortex, V2, V4. So there is topography that is transmitted. And so this is the fundamental idea in convolutional neural networks. If you have a representation of the possible arrangements that the local neurons can have, you can account for these 3 invariances: location invariance, translation invariance, and rotational invariance. I said location invariance, I meant scaling invariance first. So scaling, translation, and rotation. Affine transformations, ultimately. They're called. I don't know if you guys learned about affine transformations in image analysis, probably. So basically, is multiplying something by a matrix. Thank you for the question. Look, that's a good question. More questions, more observations. Just let me point out to you here that the 3 blue 1 brown videos also have a nice little transcript that also has the possibility of basically drawing your own numbers and playing with the networks and seeing some of these alternatives emerging here. So I just added the link into our Brightspace page if you want to do this later and you feel like you're missing it. Shall we move forward? Or do I have any more questions? I asked you to prepare 2 questions and 2 observations. It was less than that. So.
I have a question.
Yes.
So you were talking about the topology and stuff, but sometimes
we're talking about topography.
Yeah, sorry. But also in the projects, I didn't look at it in detail, but there's like situations where you flatten your data set and just make it one-dimensional when it's not. So when does that happen, when you don't care about it? topography, or what?
So do-it-yourself, in the do-it-yourself, great question. So I think that you're referring to the do-it-yourself receptive field project, right?
Yeah, I think that was the one. Yeah.
Yeah, and the idea there was to design a receptive field that recognizes oriented bars, right? And so you had 9 pixels. And these 9 pixels had to represent, to be selected for, to say, for instance, recognize a vertical bar. So in principle, when you when you send the retinal information to the lateral geniculate nucleus, there is no, so you could, in principle, have any arrangement. You know, you could have, like, you could scramble them, but that would break locality. In that project, we scramble them. We talk about the projection as being from one-dimensional vectors onto the receiving neuron. But you should think about that in the context of a convolutional neural net, in the context of arrays. In the bottom of that project, there was a notion that you would be looking into local neighborhoods in the retinal data, which would enforce topography. So the reason why topography emerges is not so much that you have to keep the stimulus unchanged, but that you are comparing local neighborhoods in that context. So the visual space, the features that you would like to recognize, say, for example, contours, they are resolved locally. Here's an example. Imagine that you are a being, a cosmological being, and you have one eye in each corner of the universe. You'll never be able to make sense of a house, right? So you do need to look at things that are spatially correlated to be able to extract features that will allow you to understand what is the object you're looking at. That's the idea of locality constraints. And then, even though you're projecting, you know, maybe a 2-dimensional stimulus to a 1-dimensional set of connections, you're restituting, you're bringing back the local topography of stimuli via the weights of the connections, via how you're combining them together in the downstream neurons. More about that in a second, but I don't know if this gave you the right picture. We'll see. But it's a good question. So, you know, if you have very abstract data, it really doesn't matter whether they're topographical or not. What matters is really the weights of the connections. All right, so if you want to say, for example, categorize a very difficult data set, what would be a good example? A sort of a, I mean, in machine learning, these come quite often. So, say, for instance, you just want to find a network that distinguishes protein introns. I don't know, and then maybe locality is not going to be fundamental. You're just going to put the sequences there, and you will find out that it's more about how the topology of the problem assembles. Maybe that actually takes us a bit far. More questions? Observations? Challenges? Want to challenge some of these notions? Okay. I hope that you can still use this even if you didn't deal with all of them. I'm not going to grade one per one. I'm going to put a set of potential answers in Brightspace now so that you can look at what my answers would be. But I think that you can also probably ask your friendly GPT for this, and he is probably going to provide very accurate solutions to it. If there are no further questions, I want to tell you a little bit about hierarchies now and invariances. and transformations. Now, first off, just to make it painfully clear what we're talking about, I mean, so these are the activation functions, you know all about them now. When you are multiplying a number, say, now, you start with a space of data, and x1 and x2. Then you apply a rotation matrix, which is the weights that are multiplying the activations. You're distorting space. So this is basically a multiplication of a matrix, right? So you're multiplying the original space by a matrix. And then you can scale it by adding a number, adding a constant. And you fold the space by applying the nonlinearities. So ultimately here, what you were seeing there in the receptive fields were how the weights impose transformations of the input space. So that's why you could see the receptive field on the later layer as a combination of the earlier layers. Each of the layers can only do this. Each of the units can only do these three operations, which are the linear algebra operations of rotating, scaling, and shifting, and then applying the nonlinearity. So that's why the spaces then become like this. I should like to point out that the necessity of nonlinearities in deep layers has to do with the fact that if you have only linear units, the combination of multiple layers is nothing but a matrix multiplying another matrix multiplying another matrix, which means you can multiply them all together, and then instead of having three layers, you suddenly have only one layer. So this is one thing that I could not go forward without telling you. So the reason why we use deep layers is because only when we have nonlinearities, there is no way to reduce all of those deep layers into a single operation. You have to first apply the nonlinearity and go further down. You can look into this proof later and note that if you have linear combinations, they're embedding linear combinations, you can substitute them all by a single one without nonlinearities. So now going to hierarchies and invariances. I said that today we were going to be discussing a little bit about the structure of neural networks. And in this case here, particularly, pattern recognition and hierarchies go super well together. And the reason is that you're slicing it, the problem a little bit at a time. You're chunking it. And we find hierarchies such as these also in the cortex, and this is the reason why we're so motivated to follow them in the artificial approaches. Here on the right side, you have the comparison between the so-called visual stream, which are, it's the visual processing layers as we know them in the brain. On the right side, goes from the retina to the lateral nucleus, LGN, to V1, primary visual cortex, V2, V3, and lock means location. So basically now you find the so-called where pathway. And you will have neurons in each of these places in the brain that will have features that are specific. So in V1 you have, as you learned, some orientation bars. In V2 you have edges and corners, which are combinations of oriented bars. In V3 you have contours, and in the lock you will say, okay, so this is to my upper right or to my left. On the left, you have in this slide you have what neurons in these different areas can be selective for. So this is a nice little summary of the types of receptive fields that we encounter in the brain. So you have in the retina photoreceptors that select for color, and so here you see RGB, and on this you have a receptive field size, basically meaning that in the retina you're really looking at pixels of the world out there. So the neuron in the retina is only getting light from a very specific place in the world. It doesn't combine a lot of layers. It doesn't have like a large spatial filter that averages things out, if that makes sense. So it's very precise. It's loads of pixels. And then in the thalamus, they already start growing a little bit. And in the ganglion cells, you would see things like the so-called on and off cells, which tell you whether you have a center illuminated and a surround dark, or vice versa, so the on and off cells, so-called. In V1, you then combine those little pixels into specific oriented layers. This has to do with the do-it-yourself receptive field project that we haven't reviewed in detail, but I still recommend you do if you're missing intuitions about how to create local receptive fields. Then you will see a complex cell, which has complex spatial features, such as, for example, a spatial frequency. You see here these little oriented bars at a certain distance from each other. This is what we call a spatial frequency. And in imaging, you might have had something along these lines. In V2, you have cells that are selective for illusory contours, textures, border ownership. I don't know if you're familiar with this illusion here, but in principle, you could be seeing either a chalice or two faces. And here, the receptive field sizes are much larger in comparison, particularly with the retina, because in order to be able to distinguish a border, you will need a large pinwheel, a large scope, and that's why these receptive fields are bigger. And as you progress into this hierarchy of processing, you then have simple shape elements in the ventral pathway, or in the dorsal pathway, the analysis of space. Where is something? Further down, you would even see complex feature configurations such as eyes and things like that. And this brings me to the point of hierarchies lead to invariances. And invariances are transformations that leave some sort of essence unchanged. We're gonna go into this now. Big paper in Nature, like 20 years ago, 17 years ago, invariant visual representation by single neurons in the human brain. It used to be a question, you know, people already knew about feed-forward hierarchical neural networks, and they thought that in order to recognize a number, say as 3, you need to have a cell in your brain that lights up only when you see 3. And that cell should light to number 3 no matter where it appears. And according to the early researchers, that led to the odd conclusion that for everything you had to recognize, you'd have to have a cell that is integrating all the features. So if you know who your grandmother is, there should be a single cell that is lighting up when you see your grandmother, the so-called grandmother cell. People said, no, this can't be. It's a distributed representation. It makes no sense. I mean, like your grandmother is a collection of activations in a large neural network. It can't be a single cell. But on the other hand, you're thinking, well, but if I'm recognizing her, like this information is somehow being summed together. In this paper, what they've demonstrated is that people have Jennifer Aniston cells and Bill Clinton cells. I don't know if you guys know Jennifer Aniston. By the time that this came out, I actually did not know who Jennifer Aniston was. But here you have a human looking at a picture, and he is getting brain surgery, and the surgeon put an electrode in his head to see, you know, where the tumor was, and they took the opportunity to show some pictures to that guy and recorded the activity of a single neuron in the brain. And here you see a raster plot, so basically spikes over time of a single neuron in 5 trials. So every row of these blue little dots here is a trial, 1, 2, 3, 4, 5. So in 5 trials that they presented this picture 5 times to the subject and recorded the popcorn, and then they accumulated in the histogram below the spikes from that picture. And it turns out that that neuron liked Jennifer Aniston, and Jennifer Aniston alone. It didn't even like when Jennifer Aniston was with Brad Pitt. And it didn't like, you know, at the time, Lakers and so Magic Johnson or some other random people. So that same neuron was only recognizing Jennifer Aniston. It also did not act to snakes or to spiders or to dolphins or to the Eiffel Tower and so forth and so on. So it had an invariant representation. Jennifer Aniston could appear with different dresses and from different angles, but it had to be her. So the semantics of the picture being shown were unique. They then did research like this for other types of concepts, and they found that there is a neuron that was responding to the Sydney Opera House. And the funny thing is that not only to the Sydney Opera House in different instantiations, but also to Sydney Opera being written. So this is not only invariant, this is obviously not only related to the image of the Sydney Opera, but to the essence of Sydney Opera, which kind of integrates not only how it looks, but how it sounds and so forth and so on. These are multimodal, invariant object cells, if you like, in the anterior cingulate gyrus. And when we look at this, you know, it's quite an astounding problem because the statistics of this are very unlikely. In order for you to, you know, how many neurons do you have to have if every neuron is responding to a single object? I mean, as many as you can recognize objects. And so it becomes quite hard to have this type of uniqueness. This paper does a very nice analysis with receptive operant curves to show that the probability of 2 neurons doing the same type of encoding was too small. So that's why the title of the paper is Invariant Visual Representations of Single Neurons. So that's the big thing. And so the brain encodes via invariances, like horizontal line. I mean, like it's basically looking at that on. So what is the same after a certain transformation? Is the meaning, for example, the meaning of an object is invariant with respect to multiple variations of the stimulus, or indeed to contrast, luminance, rotation, scaling. These are other type of invariances. You also have time invariances when saying a word now and later will have the same meaning. So that's because it's time invariant. And this is a very fundamental feature of the kinds of processing that our brains do, if it makes sense. And hierarchies are associated with this because of this idea of parallel distributed processing. In different layers, you're assembling different features. And these features are invariantly recognized with respect to variations. People tried some of these features, even sort of more abstract features. And this is an old piece of research that was searching for you know, what does the visual cortex actually represent? And they were measuring in the bottom here histograms of the probability that, say, you respond to this nipple-looking figure, but the neuron would respond with no firing rate whatsoever for just a circle. So it had to be this complex shape. So people were trying to discover what these object invariances are. And you could make little maps in the cortex to see, okay, so where are these different types of object recognizers? And it's the question is now here, how do you assemble this all? What is the meaning of all of this, right? So what's the chance that the stimulus that you're showing to the neuron is actually that what the cell is selecting for, or is this a side product of the organization of the different layers? In the human cortex, and this brings me to the question of object invariance. In the human cortex, I told you that we have these hierarchical structures, and we can recognize an object in different parts of our visual field. And the question is, how do we go about it? Fukushima invented the grandmother of the deep networks, the neocognitron, and it's basically one of the first convolutional neural networks. And a convolutional neural network is extracting a different feature per layer. In the early layers, you will see things like orientation, and in the deep layers, you will have more complex shapes. He used this network to recognize. If you did the do-it-yourself receptive field project, you will notice that the first layer is very much alike the layers that we had before. So if here on the entry point, 19 by 19 is a picture of 19 by 19 pixels containing a letter. First layer, you have a bunch of these little feature detectors, which are local feature detectors that are arrayed, covering different parts of that picture to extract what are the features and how do they get assembled. And this is why going about this way, you could then produce a picture of a T in different places of the network. And say, for example, here, you have 3 T's in this picture, and you have 3 neurons in the downstream layer that see T's. They're converging onto those. For those downstream layers, all of them are recognizing a T. If you want then a grandmother cell or a T cell to recognize all of the 3, you need to have a third layer that's receiving from all of the T-like positions that are local into the conclusion or into the output unit, so forth, so to speak. So this, by going about this way, now this in this picture here you can see, so by going about this way and having individual layers in which features were being disassembled first and then reassembled next, you could have invariant recognition of a shape such as a letter. Or a number like this. So in here you have the first layer being presented 19 by 19 pixels with the number 2. Then the first layer has the feature detectors. The first feature detector is horizontal line, then like slanted, like diagonal. And you can see how they are basically extracting local information. Basically, every one of these features here is asking, where are the horizontal lines in the input picture? And to then assemble these in the later layers. The main idea, and in fact, I don't know if I have this as an animated version of this, but maybe you can get the idea here of what we're doing. So each one of these filters is looking at the entire picture and creating an output which assembles the answer of looking into each of these smaller scope pictures. So here, for instance, you have the number 2. This is and then you have a structuring element or a convolutional filter that looks like this. So the convolutional filter, apologies, struggling. So the convolutional filter, which is the weights of your network looking at the particular place of the image, could have these weights: negative to the left, positive to the right, and 0 in the middle. If you now convolve the picture with this structuring element, you will see the places where the vertical lines are, and then they score high, and they are a contrast. And then you can see here how from the original picture, you would have this resulting picture here, which gives you positive to right edges and negative to left edges. I'm assuming that that a lot of this already comes for you from image analysis. If you haven't had it before, then this might be a bit too confusing. But this is what I wanted to tell you. So hierarchical neural networks, the idea is that you are convolving the earlier layers with filtering elements that are using local information and then reassembling them in more complex features, such as a classifier. This is just an elaboration of what you may have seen in the 3 Blue 1 Brown videos. Stop now and ask, do you have questions?
I have a question.
Yep.
So you were saying like everything you can recognize is like a single neuron, but couldn't you like use less neurons to recognize the same amount of things overall? Like you don't need 10 bits to represent 10 numbers. Like what?
I mean, you can definitely use compression, right? So what I was saying is that the argument that Koch leveraged when he was publishing that paper, and this is basically about his argument more than what there is. So the argument in this paper was that they find evidence for the representation of very highly abstract concepts landing into single neurons. It doesn't mean that you couldn't have it with more neurons. It means that they have found single neurons that correlate with highly abstract entities. This is not, yeah, like ultimately I would have to talk about latent spaces. So ask away, and maybe it can help. Sorry, Boris.
I forgot what I was gonna ask exactly. I was gonna ask, like, so is there literally like one neuron per one concept? Like, couldn't it be that in that
I think the long answer is we don't know. The only thing we know is that we're getting more abstract with larger receptive fields the further we go. And we cannot have infinitely many or as many neurons as we have concepts. So we still are at the level of this this paradox. So there's still paradox, and we don't know, because we, in order to be able to tell for sure, we would have to measure all the neurons, and it leads to funny ideas, right? So say you have your grandmother, and I go and find that neuron. If it's only one neuron and I burn it in your brain, do you forget your grandmother? That's the big question, right? And it turns out that you can lose very specific memories, like with very localized lesions. Only that it's really hard to find, because how do you know what you don't know? So there's a lot of these epistemological problems in these types of research. But this is how we use artificial neural networks in the context of computational neuroscience to try to understand what is the encoding capacity of the brain. So the spirit of this lecture is this: we're using artificial neural networks as representations of the types of processing that we see. Now, there's many differences here. For example, our networks are not fully feed forward. They're also recurrent. So the networks project forward and backwards. And we're going to talk about that in the next moment of the lecture now. Okay, shall we have a pause? Shall we have a pause for 15 minutes? Let's meet back at 4:05, okay? And I'll tell you about learning and plasticity. It's going to be a slide fry, as you know, but I hope that it will give you the basic elements of plasticity. So 4:05, we meet again. I'll resume speaking, and if everybody is with me, is great. If they will come later, they can see my summary offline. So here we go. Last block. I know the suspect, guys. I mean, like, I just want to, the way in which this course is conceived is about the foundations. So there's a lot of interesting papers that bring a lot of these concepts together, but I feel like if I give you the right terms, you will know how to search for it yourself, and you will be better off when if you're opening the literature, opening vast Pandora's box of this type of literature. And because of that, I'm going to tell you about plasticity in the last hour of our time together. And I will start by reminding you that there's a difference between learning and plasticity. Then I'm going to big taxonomical changes that are related to artificial neural networks. I'm going to define them, and then I'm going to just basically say that in the brain all goes together as a big mess, and it's hard to tell which one is doing what. But I'm still going to give you some examples of each in the context of artificial neural networks and computational neuroscience. Then I'm going to tell you the number one dogma about brain learning, which is synaptic plasticity via Hebbian learning. And then I'm going to tell you about its spiking version, which is spike time dependent plasticity, or STDP, and another homeostatic learning rule called BCM, Binnenstock-Cooper-Monroe. And then I'm going to give you a few examples of learning with plasticity in the context of receptive field formation. Moving right on. Synapses are changing all the time. Biology is variable, and the synapses, sometimes they grow, they shrink, and they're immensely complex, as we talked about. But the point is that learning is changing the synapses in a particular way as to make them more meaningful or useful in the context of the processing in the network. The types of procedures that exist to train synapses in artificial neural networks have actually come back to biological neural networks as metaphors. The extent to which the metaphors apply is always subject to very careful analysis. But it's good that we have these terms so that we can start discussion. What the example we had so far was artificial neural networks, and that with backpropagation. And that is a standard example of supervised learning. Errors are very strictly defined. That what is not what you wanted to get minus what you got is the actual error. There's always a cost function, it says. You've erred by this much. That means there's always a gradient. And so you use backpropagation to subtract, change the weights in the direction that reduce the error. In unsupervised learning, which is way more interesting from the perspective of biology, there is no true error. We don't know what is right. It's just basically changing as a function of activity. It uses correlations in the activity of neurons to tune the connections between those two neurons. It uses activations and co-activations as a measure of how strong neurons should be connected together. It leads to self-organization without having to define a priori what errors are. If you're a philosopher, or if you're philosophically inclined, you would know how difficult it is to define what an error actually is. And if you're a biologist, you're thinking, well, we are mud that stood up. So in principle, there is some self-organization that has bootstrapped our ability to tell what matters in the world. And so that is through self-organization. And particularly if you have a biological sensor that tells you that something feels good, consciousness and subjective experience here notwithstanding, you have a success function. So say like I'm hungry and I eat, and I get happy, this is a reward function. So I'm going to do the thing that I've just done. So if now I like chocolate, and I know I just go to the cupboard and get chocolate, and I eat chocolate and I'm happy, then there's a chance that I'm going to eat more chocolate, and I'll learn, reinforce that habit over and over until I become morbidly obese. So unless, of course, I have some other competing rewards. So it's interesting now to think about exploitation versus exploration in that context. There is a reward function, and that reward function is built in us so that we can recognize what the situation was. And there is a memory trace, the thing you just done. So in reinforcement learning and reward learning, the idea is that if something went right, then the things that you did that led you to that are the ones that you're going to be reinforcing. I'm not going to talk about reinforcement learning a lot here at all. I'm going to tell you more about plasticity and learning in general, but I wanted to create these three big pillars so that you understand what we're talking about. Primarily today, we already talked about supervised learning. Now I'm going to tell you about unsupervised learning, primarily. To talk about reinforcement learning, there are a lot of videos out there, and you can also take that as a topic for your final project. Say, like Mario didn't tell us a lot about reinforcement learning, I'm going to tell you a little bit about reinforcement learning. I remind you that the closer to biology, the better. Okay, so it would be nice to look into eligibility traces and three-factor rules. A couple of these things are in the spreadsheet that we talked about. That is now in Brightspace linked. You can have a look at some suggestion topics. One of them is that. Now let's move right on. How would you start from nothing and think about a self-organization rule that would apply to all neurons in the brain, such that you would learn about things in the world? Donald Hebb was a psychologist, and he thought that one of the most astounding facts about human cognition is that we are associating things all the time. And so, yellow bananas, I tell you banana, you think yellow. I ask you a color. And it's like a priming examples, for instance. But there's a lot of the correlations that happen in the world that we're learning, the laws in the outside world, which are of David Hume's type. So are about apposition of ideas. And the basic form of Hebbian learning, and Hebb came up with a postulate for that. He didn't call it the Hebb's postulate because he was not, he was more humble than that, but nowadays we call it the Hebb's postulate, which is neurons that fire together, wire together. And so the change of the weight between two neurons, AI and AJ, is proportional to the co-activation of those neurons. If neuron AI participated in the firing of neuron AJ and neuron AJ fired, then you should increase their weight. And this is the mathematical formulation for that. What's wrong with this? What's wrong with this? I ask you. Nanobiologists, stop now and look at this formula and say, like, what's the problem with this idea?
Sounds like it's going to feedback loop and keep increasing.
Yeah, forever. So it runs, it doesn't have, it's unbounded. Mathematicians don't like unbounded things. Now, unbounded Hebb rule doesn't work for most purposes. You need to create some sort of normalization of the weights. You need to somehow inspect how they're all contributing to the firing of the postsynaptic neuron. And one possible idea to stabilize Hebbian learning is to subtract the average activity of certain time preceding. So say here you have eta, which is a learning rate. I didn't tell you that in the previous slide. The eta was the learning rate, is an amount by which the correlation updates the weight. So like I just go back into the previous slide. So this eta is the efficacy of weight change. which is the amount by which the correlation between the two neurons is transformed into W's. So if you have neuron 1 and neuron 2 firing at maximal firing rate 1 Hertz, then an eta is 0.1, then you would add to their weight 10%. Now here, AI bar and AJ bar are the averages of the activities of neurons AI and AJ. And here you would have to define, okay, how are you computing this average? Is it the all-time average, or is it like only the last 5 seconds of activity, right? So you have to make commitments here when you're stabilizing these rules as well. Another way to stabilize it is to subtract the weight contributions, to try to normalize by the weights. Delta Wij equals the correlation minus the, you should think of this as the dynamic range of the neuron, is Wi times Aj. So basically, by how much can the entire Aj activity go to? So this is actually a mathematical insight of some sophistication, and I am not going to break it down to you the entire proof, but I will tell you where you can get that. In 1982, there were a few ideas related to unsupervised learning that were really fundamental. One of them is that neurons they become principal component analyzers. They're able to find the plane of maximal correlation between activities of two neurons. Now, if you think this animation that I have here running in the bottom, you have the activity of neuron 1 and the activity of neuron 2. Now, you will notice that these two neurons covary, right? There is a way, so by applying this learning function, Oja's rule, Oja being the Finnish person that learned it, which will spot the plane of maximum correlation between any two given neurons. These ones happen to correlate perfectly like this, but they could also be anticorrelated. The idea here is that, and let me confuse you a bit. Here on the left, this formula is the formula from the paper, and the reason why the terms are all different from what you've seen before is because I want to get your attention to the fact that in our field, people use a vast variety of nomenclatures, which might confuse you. In this case here, eta is the activity of the postsynaptic neuron, mu are the weights, and xi's are the activations. So this is basically saying A, the activity of the neuron, is the sum of all the weights times the activity, the linear combination. The same thing as we had for the artificial neural networks we've been handling so far. And the change of weights, that is, by how much the mu will change in the next time step, is a proportion to what we had before, plus a gamma here, which is the learning rate, times the eta t, which is the activity of the postsynaptic neuron, times the psi t, which is the correlation term. If you notice, this is basically AI times AJ, divided by the normalization of that process. If this is not immediately clear, it doesn't matter. You can look into the paper to learn more. The important thing that I would like you to remember is that this doesn't work. You have unbounded growth of weights, and so you have to find a strategy to get the way to keep the weights bounded. And the strategy comes from this paper, Oja's rule, simplified neuronal model as a principal component analyzer. You will notice here that this neuron doesn't even have a nonlinearity, right? It's interesting. It's just basically a linear combination of weights times activations of the presynaptic neurons. Another paper by Finnish guys, 1982. Like a lot of Finnish guys thought about interesting things. So this is the idea of self-organizing maps. It uses Oya's rule to compute correlations between local field of activations. And it's an interesting paper where they see the emergence of receptive field-like structures, such as appear in V1. Here is another interesting fact. Everybody do this now. So put a circle, make a circle with your hand here. Now choose a point in the world. So it could be some object, like I don't know where you are, but choose an object and look at it through that hole. Now, if you're able to blink one eye and then blink the other one, you will notice that the object that you were looking at is only through your hand in one specific eye. That's odd. So it's because, you know, the two eyes are separate, so you have disparity to see 3D, but it also means that the image that you're forming in your brain is more related to what the image of coming in a particular eye. So this is called the ocular dominance column. So you have ocular dominance. And in your brain, you can find neurons that are responding more to one eye or neurons that are responding more to another eye. So your ocular dominance actually also flickers, which is interesting. But I wanted to tell you about this third rule here of normalizing neurons, which is called the BCM rule. So each eye is represented in an organized way in the cortex. You have basically, so this is how the information, the visual information propagates through the optical nerve. You see the peripheral vision here in the temporal retina mapping onto one side of the cortex, peripheral vision of the other side mapping to the other side, but then they mix in the primary visual cortex, say in layer 4, which is the granular layer of the cortex. So-called binocular rivalry. One question in science had been, how does this pattern of receptive fields form? We found these receptive fields, they're there in cats, they're there in humans. How do they form? People made experiments in which they took newborn cats, closed one eye of the cat, and then looked at the brain to see how these receptive fields would self-organize in the level of the cortex. And they noticed that depending on where they closed the eye, when they closed the eye, you had different structures forming in the visual cortex. Basically here what they're saying is that in the beginning, in the normal situation, you already have a little bit of baseline, ocular dominance columns. So this blue and green here represent the receptive fields for one eye and the other in the cortex. If you close the eye of the cat at birth, and then you measure it at different times, you will notice that the ocular dominance columns occupy the visual cortex in different ways. If you close it at birth, one eye takes over the entire visual cortex. If you close it 6 weeks later, much less of this reorganization occurs. Point being, the binocular rivalry there separates the cortex into tessellations, if you like, that look a little bit like left eye, right eye, left eye, right eye, and so forth. The BCM rule, it is a system to normalize weights in unsupervised learning. It's a system to correct for the Hebbian's problem of unbounded learning. And in this case here, let's look at the equations. Y is the activity of the postsynaptic neurons, say one neuron in your visual cortex. Sigma is a saturating nonlinearity. For example, a sigmoid function, 0 to 1. Then you have the summation of the activities of neurons xi times their weights wi. You impose onto them a plasticity rule that changes the weights over time as a function of the postsynaptic neuron activity y and its comparison with theta m, which is the average of recent activity of neuron y. So you're saying, if you're more active than the average activity you had before, then you should enforce your learning. If you're, as a function of the correlation with the input, xi. But if you're not sufficiently activated by that neuron, you have a chain. If you are too much higher than the average activity you had before, that change is positive. If you're, on the other hand, if your activity is lower than your average activity, your change of weights is negative. Then you have here an xi term, which is the correlation term. So x times y, so xi, presynaptic neuron, y, postsynaptic neuron, multiplied by each other, the correlation. Y minus theta, a direction, plus or minus. Sigma prime of y is basically just the derivative of the function. There is an analytic reason for this to appear. And theta m is then the expectation of the recent activity, right? So you basically land with a rule that looks a little bit like this here on the left side, in which the change of weights, now represented by phi y, is a function of the average activity theta y. If y, the activity, is larger than theta I, the modification is positive. If it is smaller, then it's negative. With this rule, they've been able to explain the dynamics of the creation of binocular rivalry columns, and it is the reason why I'm mentioning the BCM rule. It's a homeostatic rule. It keeps weights bounded in any case, and you can read more about it in Scholarpedia, or even do this as a little side project. Ask ChatGPT for a friend for help. There are many more examples of these types of nonlinear receptive field formation with homeostatic type learning rules. People have been able to observe receptive field formation such as in the visual cortex, in hippocampal place cells. You might or might not have heard about place cells before, but these are cells in your hippocampus that are active when you are in a particular place of the environment. People have been able to see this with BCM rule variants. People have been able to see spatial map formation with Hebbian type rules. The emergence of the emergence of topographical maps in the cortex, such as the somatosensory maps, can also be explained by Hebbian type rules. So this is an early computational model where they show that by cutting off a finger and getting rid of those neurons that use that receptive field, eventually Hebbian learning rules makes the other fingers occupy the available cortical space. This actually has been observed in primates, and that's the reason why I show you. This is just for you to have an idea how powerful unsupervised learning rules are. I mean, you might not understand all of the details. Here's another paper on this receptive field formation and formation of topographic maps in spiking networks. And before I leave you, I wanted to tell you that Hebbian learning has a spiking version, which is called spike time dependent plasticity. That doesn't take simply the activity of two neurons in their firing rate, but how close by each other two spikes happen. This is the main paper. What they've shown here is that by pairing the activity of a postsynaptic neuron, the after neuron, if you like, with a presynaptic neuron with a particular interval, they can potentiate the connection between these two neurons. And for very far away spikes, that does not happen. This can be summarized in something in this graph. So here we have in the x-axis the difference in spike timing before the pre and the post synaptic spike, and in the y-axis you have the change of weight as measured by the amplitude of the excitatory postsynaptic current in the receiving neuron. So you see that when the postsynaptic neuron follows the presynaptic neuron, that is spike timing is positive, the change in EPSC amplitude is positive, and vice versa. So this is the essence of spike time dependent plasticity. It basically embodies Hebbian learning in certain ways, because the idea that neurons that fire together wire together here is basically the neuron that fired before. If the neuron fired before led to change in the neuron that fired afterwards, you're going to potentiate that connection. The molecular biology of this process is really interesting. It has to do with AMPA expression, but it gives you the sense that spikes matter. It's not only firing frequency. You can also demonstrate these things with spiking rules. I have one more slide about STDP, which basically tells you that in different animals, we found different types of spike dependency plasticity rules. And in this case here, unfortunately, because the literature is annoying, they flipped the temporal axis, and now they are measuring T, the time of the pre-spike minus the time of the post-spike. And if that's positive, that means that pre came after post, right? Now you can then see the different instantiations of LTP and LTD. And you see cases in which you, for example, in GABAergic neurons, hippocampal culture here in D, you'd have something that is very close to typical Hebbian learning. Just simply when two spikes are close by each other, doesn't matter if they pre or post, they fire together. That's a mechanism of cluster formation. And in the case of E, the neocortex layer 4 spiny stellate, you have cells that hate to fire together. They don't like to be synchronous. And so this is a method of separation in neural networks. We basically make larger neural networks like this, and we observe then some biological principles of computation emerging from these plasticity, unsupervised plasticity rules. Conclusions, I mean, the first is not even a conclusion, it's the premise with which I started. Learning depends on plasticity, but that's not the same thing. Hebbian rule is unstable, needs to keep weights bounded. There's some few different strategies to do so. There's Oya's rule. There's BCM, which is a homeostatic rule. There's spike time dependent plasticity, which is spiking Hebbian learning. And I wanted to insist that Hebbian types plasticity leads to development of interesting receptive fields. That is, we can explain some features of cortical organization via these types of unsupervised plasticity rules. All right, it's 4:35. The two things we have to do now. You have to ask me questions if you have them for this bit. I mean, I can also point you to more resources. And then I'm going to tell you to click on the spreadsheet. But first, questions. Questions?
I have a question. So when you were talking about the thing where they were looking at someone who lost a finger, and then the what did you call it, cortical space or whatever, took over.
Yeah, real estate. Some people actually have called it cortical real estate.
So I was thinking back to the thing I saw. Well, I don't really remember the details, but the gist of it was they were making like an artificial second thumb, and people got used to it surprisingly quickly. And I was wondering, like, would that take the space from the other fingers then, or how would that work?
What a great question. What a great question. I mean, it really depends a little bit on the brain-computer interface that had been used here. In principle, thinking about something is a really free thing. You can think about moving the finger you don't have. Say right now, right? Like, look at your hands, like just think about moving a finger you don't have on the right hand. Now think about moving a finger that you don't have on the left hand. You can do that, right? Most people can do that. I mean, like it's your phantom finger. And in a sense, what you need for the brain-computer interface to read is the thought. If the thought can already be concocted even before the finger exists, then you don't need new receptive fields because you're just using the proxy. However, if now you give this finger to the person and you start to pump in the sensory information as feedback to the cortex, there's likely that there's going to be some reorganization. And in that case, I wouldn't be surprised if they will be using some space of the other fingers. Your toes are actually not very well separated at all in your cerebral cortex. So it's kind of like a blurb of multiple fingers, and the middle finger of the toe, or like the index finger of the toe, they move together kind of, and they're highly correlated. We don't use them separated as much, and so their receptive fields contaminate each other. So you measure, tell people to measure their left finger or measure their left finger, and the receptive fields of the surrounding fingers will be active. In the hand is less so. So the thing here, I think that the main message is that correlation will determine, or co-activation of different sensors will determine which patches of skin, for instance, or which muscles will be co-encoded. It's also interesting to think about motor cortical areas, because there you don't have isolated muscles, you have muscle synergies, things you can do with multiple muscles at the same time. So you also don't have specific muscle receptive fields, you have more like movement receptive fields, if you like. So the thing that the arm does. And those are more or less well structured in the motor cortex. We're going to talk about this in NB3014. If you're taking primary neuroscience, it will be fun, because this is the kind of model that underlies our current understanding of these observables of science. Good question. Thank you, Boris. Other questions? Yeah, I mean, I know that this is super compressed. You didn't really have the time to unpack each of them, but I just one more time, and for the last time in this course, I'm going to tell you, this is just for you to know that things exist. Might go back to the slides, might stumble upon it in the future, and I hope that it will be useful elements of a potential understanding that you're building in the brain, okay? About brain things. So that's the spirit on which it's given. That's why sometimes it's more than what you can bite. Each of these certainly can unfold into like entire lectures, right? You have to do the bifurcation, to do a lot of analysis to make these things to make these things formally clean. But if you know they're there, then you might be able to find them. You're always able to find me back, say like, Mario, when you mentioned this, I mean, did anyone have a proof for that? What's the main paper for this? Or like, I'm going to this lab and they told me about this phenomenon. Is there a model that can be used for this? I'd be happy to answer any of these questions in the future. Just find me in the lab, like at the 12th floor, or strike me an email, things like that, okay? Irrespectively. In any case, this is all I had for you today. Now, the last thing is right space. You can go to, for the student presentations, for the projects, I've updated this link here, and now it says you can see some examples topics in this spreadsheet. If you pick a topic, comment on that topic with your name or and your partners so that others know what you've picked. So this oh, it's double, the HTTPS is doubled. Great. Let me actually edit this now. Hoping that you could already figure out that the link had this oddity. How do you edit the link here? By clicking this, hopefully. The URL is not valid.
I think you still need a semicolon, or what is it?
There's a colon missing from the HTTPS.
Yeah.
Oh, sorry, you're right about that. Thank you. Okay, click. All right, so you can, so this should be free to comment. So you could probably click your right mouse button here, just comment and say like, I want to do this, just so that we maximize the spread of topics. You don't need to pick up any of these topics. Just have a look at what there is. I'd be happy with any topic. If you have a different topic, you can also add here as a comment, and then I'll update the list to include your topic. So I did put some goals here that might help you orienting your pursuit, and some description of the project. Maybe there are some references and so forth. Any questions?
Yeah, just two basic questions about the presentations. If we come up with our own topic, can we shoot you an email to get your opinion on it? Is that fine?
Yes, you can sanity check with me, with the caveat that I'm busy. I mean, like the quality of my answer is going to be probably underwhelming. I'm going to write you a few sentences saying yes, no, this direction, maybe.
Okay, and the second, do we need to upload the presentations in, or do we just show up with them on the day?
You show up with them on the day.
Okay.
If anyone cannot make to the day, it's okay that you do a video, okay?
Okay, thank you.
So anything counts as a report, basically. We're going to be using peer reviews for the presentation. The rubric is here. So this is presentation slides, and there's like everything together, right? So it's a project. I do appreciate that you describe what you're going to do well, right? It's like there should be at least 1 or 2 paragraphs of very clear-minded, very topical. It's like, this is what I'm going to do. Scoping. Scoping is important. And it's nice to have a little bit of originality as well. Originality in this case here simply means your own insight. What you thought was interesting to pick from something. I mean, like some selection can also, how you select, how you put them together. Okay. And then I'll get some slides. Once you have this, I'll update this, and then you can see more or less, I mean, like how it's going to look like. So these are the times, and there are going to be some breaks, and depending on how many groups are there present, we're going to cover the rest of the day. Is that all right? Okie dokie then. I have nothing else for you today. So if you have nothing else for me today, then I shall love you and give you. See you next week.
Did have a note. We can't add our names in the project because it's comment
You
only.
can only, as a comment, you should be able to. So you have to comment. like this. Myself and I. So and you should be able to comment, I'm hoping. You have commenter rights. Anyone with the link is a commenter. I don't want you to edit the main things on the text because I cannot freeze specific parts, and so you just basically like people did here. Look, Boris already basically locked calcium channels. Lovely.
I heard a short question
Yes. Marianne.
about the grading. Is this a pass-fail course?
No, it's going to be graded, so I cannot make it pass-fail because of what's in evals. So we're going to grade it like this, and there will be a peer review tool where you can then ascribe values to your peers. In the future, it should be a pass-fail course. I'm going to change it to do that, but for the moment, I'm doing this still. Thank you for the question. Is this answered, Marianne? Is this clear? Do you have some observations, or would you like to give some feedback?
It is clear we had some issues with speakers because like we turned them on and you had like this echo
Yeah. Like there's feedbacks all over. Makes sense, yeah.
because we're using the board.
It makes sense. Okay. We're good. I wish you lots of fun preparing it. Use ChatGPT. Just be sure to disclaim what's your own work. I'm happy that you assemble this in your own way. Make it rich, make it fun, and reach me if you need me. Okay.
Yep.
Have a good time, everyone, and we talk soon.
Thank you.
Thank you.
Bye.
Thank you. Bye.
Bye.